Last time, you will recall that we were talking about the roles of insulin and glucagon in controlling glycogen metabolism as well as glycolysis. So we looked at the well-fed state, and of course, when we're well-fed, we're in the business of taking our glucose and making glycogen or using it for glycolysis. We're not in the business of glycogen breakdown, for example. It's all very logical, even though at the same time, you know, there's a lot of different pathways and a lot of things going on. The overall picture is actually pretty straightforward. So, as is true, this overall picture also holds for between meals and fasting. We want to break glycogen down. We want to prevent glycogen synthesis, and we want to prevent glycolysis because we don't have large sources of energy. We can rebuild some sources of energy by breaking down glycogen, as well as other things in our body. But right now we're focused on starches and sugars and things, right? So what's going to happen in this case? Well, glucagon is the hormone that will be signaling through that cascade we looked at, signaling through a receptor, a cell receptor, to raise the levels of CAMP. That in turn will raise the levels of pKa, and that will do a couple things. That will increase the phosphorylase kinase activity that gives us the glycogen phosphorylase A form, the active form that stimulates glycogen breakdown. This kinase activity will also prevent glycogen synthase from acting. Remember, when glycogen synthase gets phosphorylated, its activity goes down, right? It's an inhibition for glycogen synthase to be phosphorylated, while it's an activator for glycogen phosphorylase to be phosphorylated. So that does two things at once, right? It increases glycogen breakdown and it prevents glycogen synthesis. We also reviewed yesterday this whole sequence of events where if we raise, actually in this case, we're between meals or fasting, if we lower the concentration of fructose 2,6-bisphosphate, we actually inhibit glycolysis, right? And that happens through this bifunctional enzyme, PFK2 and FB-PACE2. And remember, when this is phosphorylated, what happens? The FB-PACE2 activity goes up, right? And anytime you see that PACE thing, that phosphatase or PACE, that in general means we're dephosphorylating this compound, right? And we're lowering its concentration. That in turn, this is an allosteric effector. Just to remind you of that, it's an allosteric effector that acts on PFK1 of glycolysis. So it inhibits that and lowers glycolysis. This also has an impact on another enzyme in the glycolytic pathway. This lowers the activity of pyruvate kinase and further inhibits glycolysis that way. Okay, so that's the whole shebang, really. We can look at the grand summary here. Well, let me just see if I'm missing anything here. I just covered all these bullet points here, right? Basically. Liver produces glucose 6-phosphate by glycogen breakdown, yay, and by gluconeogenesis, which isn't really represented in this figure here. Stops using glucose to fuel glycolysis or to make glycogen. Okay, we want to just give some consideration here to liver versus muscle. and how these organs and tissues differ. So muscle is a bit selfish. It uses stored glycogen only for its own needs. So, and this makes sense because the muscle undergoes very large changes in its demand for ATP under, intense exercise or whatever. It doesn't have an enzymatic machinery for gluconeogenesis, as does the liver. It responds to epinephrine, which will increase glycogen breakdown as well as glycolysis and restore ATP. So the liver, however, is in the business, as I've mentioned several times now, of responding to organism-wide needs for glucose. And so it will actually not just use glycogen for its own needs, but it's very much in the business of replenishing blood glucose as needed, which is what we see here. So we know this can happen through the breakdown of glycogen or through gluconeogenesis, and it can equilibrate things as needed depending on whether the bloodstream is rich in glucose or needing glucose, whatever the prevailing conditions are. Yeah?
So is the goal in the liver and compared to the muscle to maintain blood glucose levels, where in the muscle is to feed as much glucose as possible to glycolysis by breaking down glycogen?
I think the main point here, hopefully this answers your question, is that the main difference here is glycogen. The liver can use glycogen in two ways. It can use it, just for glycolysis to supply the energy needs or whatever of the liver. But it's also important, has this important role of helping maintain blood glucose homeostasis, right? Whereas glycogen, whereas the muscle is just, we're kind of talking about the glycogen level right now more than the glucose level, but muscle is never going to export glucose, if this answers your question, the way the liver is doing. Okay.
Before the glucose 6-phosphate can into the blood glucose, it has to lose a phosphate group, though, right?
No, there's actually a G6P transporter. But actually, I think you're right, though. I think there is a G6P transporter, but I think typically it is dephosphorylated before it goes through the There's a whole host of glucose transporters, but I think it goes out through the GLUT2 transporter or something.
Do we need to know all those glucose transporters?
Not really. I think at one point I distinguished between GLUT1, 2, and 3. I'm not remembering off the top of my head, but I'm not going to make you go into detail about glucose transporters. Okay, this is here for you. I'm not going to sit here and read through all this. This is just a recapitulation. Again, I took this largely from the book with, I did my own highlighting and modifications here to, make this hopefully as clear and explanatory as possible. So, but it does, I don't want to overemphasize this, but I do think it's really important you can basically, from understanding the needs of the body or the organism or the cells under either well-fed conditions or fasting conditions, you can predict the activity of any given enzyme, right? Whether it be glycogen synthase or glycogen phosphorylase or glycolytic enzymes or whatever, because the logic is just there. And again, that kind of logic is breaking down glycogen in the fasting state, engaging in gluconeogenesis, yes. Inhibiting glycogen synthesis, yes. Inhibiting glycolysis, yes. Just that's, and then for any given enzyme, you can think about that and say, well, this enzyme must be inhibited or this enzyme must be activated. And you do need to know whether phosphorylation activates or inhibits that particular enzyme, but it all unfolds from, as I said, I think last time I used the term first principles, it all unfolds from what your body needs. It really does. And that's, I think, all explained here. So this is the end of chapter 15, I think. And now we can go on to chapter 16. Which is the citric acid cycle, but we're gonna cover some territory that leads into the citric acid cycle and give the overview of the whole cellular respiration process as well. So yeah, before we, hang on, let me share the slides here. Actually, I think I just want to share my screen. Yeah. Before we delve into cellular respiration, before we delve into the citric acid cycle, we want to look at this overall process that is known as cellular respiration. And this is the process in which we take the pyruvate that is produced in glycolysis, and the whole big thing is we oxidize fully this pyruvate produced from glycolysis to H2O and carbon dioxide. And the ultimate goal is not only to go through the citric acid cycle, but to go through oxidative phosphorylation, which is a chain of electron transport carriers, and make a whole heap of ATP. So that's the big picture. So if we're talking about glucose, fatty acids, and certain amino acids, we get acetyl CoA. So this is a 2-carbon fragment, the acetyl group, attached to a very large coenzyme, coenzyme A. And this is an activated compound that is ready to go into, you guessed it, the citric acid cycle. So this oxidizes these acetyl groups to carbon dioxide, and along the way, energy is, and the energy that is released is actually conserved in our reduced electron carriers, NADH and FADH2, which will go on to this electron transport chain into oxidative phosphorylation. And this is the money maker. This is where we make a whole bunch of ATP. Okay, so how does this look in a more visual way? Well, our main focus, we're going to talk about the catabolism of fatty acids. We're not really going to talk extensively about catabolism of amino acids, although we will give some attention to ketone bodies. So today we're looking at, we've already been through glycolysis. We're going to pick up here from pyruvate, and we're going to spend a good amount of time talking about this extremely fascinating and large enzyme called the pyruvate dehydrogenase complex, which is going to decarboxylate pyruvate to form this acetyl CoA. It's actually also going to generate some NADH right here. This is formally not part of the citric acid cycle, but neither do we treat it as part of glycolysis. It's like sandwiched in between the two things. So from there, as noted, we can imagine acetyl CoA getting fed into the citric acid cycle, which has different names. You may have heard TCA cycle, you may have heard Krebs cycle. It's all the same thing. It is a universal pathway, much as glycolysis is. And we're talking about the oxidation of the acetyl groups and the generation of these reduced electron carriers, NADH and FADH2. So this is, a big part of extracting more energy from the glucose molecules that we started out with at the beginning of glycolysis. Stage 3 here is the these reduced electron carriers being taken to this electron transfer chain. Actually, oxygen is the final electron acceptor here. And through the courtesy, we'll be talking about this in a lot of detail, but the electron flow actually sets up an electrochemical gradient where we get a lot of protons on one side of the mitochondrial membrane, and as they flow back through, this actually powers the synthesis of ATP. So it's not the electron flow does drive it, but protons are involved as well. Okay, so that's the big picture. 1, 2, 3, bada bada boom. Lots of details to come. So, production of acetyl CoA or activated acetate. This is all about the PDH complex. So, oxidative decarboxylation, we have seen it before, you may recall. We've seen it with pyruvate decarboxylase, and we've seen the transfer of carbon dioxide with the transketolase reaction, right? We've seen a 2-carbon moiety being taken away from one molecule and added to another in this transketolase reaction. Well, if we're going to get from pyruvate, you may recall, if we think back to the anaerobic conditions, we could take pyruvate, and if we're getting to ethanol, we can lose this group to form acetaldehyde. That's what happened in the first step of making ethanol under anaerobic conditions. In this case, we're going again to remove this carboxyl group here, and it's going to end up being attached to coenzyme A. We're going to look at the structure of coenzyme A in a minute. In this case, unlike the anaerobic conditions we had for the ethanol fermentation, here we have an oxidative decarboxylation. So we have oxygen available. This is basically an irreversible process. We do release a CO2 in the end, and we generate some NADH. This is called the enzyme complex, and it truly is a complex. We'll look at the structure in a moment, is known as the pyruvate dehydrogenase complex, and it's actually 3 enzyme parts stitched together. It's a cluster of enzymes and these cofactors. And this is the first time we sort of up to now have tended to gloss over the cofactors a little bit. You know, I may have mentioned that they're essential, but we haven't really looked at their structures particularly, and we haven't looked at their chemistry very much. So we're going to delve more deeply into that today, because we'll see how incredibly important these things are and really why. Another thing to take note of is this is all taking place in the mitochondrial matrix. So glycolysis was in the cytosol, and this is happening in the mitochondrial matrix. So we have to get pyruvate in there, right, as we talked about previously. Okay, so the PDH complex itself involves a dehydrogenation and a decarboxylation. And these are the 3 enzymes at play: pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. Conveniently abbreviated as E1, E2, and E3. So the 5 cofactors here, and in particular, these first 3, we're going to see, as mentioned, how important they are. We've got thiamine pyrophosphate TPP, lipoate, sometimes called lipoic acid, coenzyme A, abbreviated as CoA, And then we have our electron carriers, FAD and NAD. It is worth noting the enormous nature of this PDH complex. Not only is it 3 different enzymes, they have many copies. So E2, the transacetylase, has 24 to 60 copies at the center of this enzymatic complex. The E1 and E3 surround all this and themselves have multiple copies. This makes this whole complex larger than a ribosome. So this is big. And we're going to see that PDH is similar to an important reaction in the citric acid cycle, which is catalyzed by the alpha ketoglutarate dehydrogenase, very similar chemistry. We're not really going to go into the structure of the alpha ketoglutarate hydrogenase, but this is a kind of conserved mechanism. We see the things that are happening here happen elsewhere in decarboxylations. Okay, so what is this TTP? So this is the structure of TPP, and it's this thiazolium ring here that's going to be doing a lot of the chemistry. So this is thiamine. This whole thing here is thiamine, and what differentiates it from thiamine is the addition of these phosphate groups. We did, I think in the last lecture or the lecture before, I did refer to TPP as addressing the problem of the carbanion. Because if we're going to decarboxylate pyruvate, you can imagine that we might have a very unstable intermediate based on the chemistry here. But the truth is this carbanion is almost never observed. So I'm going to show a video on some of this chemistry because I found it recently, and I think it's, even though I'm not, by the way, I'm not going to hold you responsible for the mechanism of all the mechanisms of the PDH complex. I want you to more have a big picture view of what's going on. But this video I found is by a true organic chemist. I do not claim to be a true organic chemist. So I think this video is interesting, even if I'm not going to hold you responsible for every, detail of the mechanism and all this very organic chemistry terminology, things like elides and, like all these kinds of things. But it's worth, just for the interest of it, it's worth knowing about. So one of the things that this TPP does, right, is it stabilizes any potential carbanion intermediate by forming this thing called a hydroxyethyl TPP. And we do have a nucleophilic attack on this carbonyl carbon here. And after, a series of arrow pushing events here, we get the decarboxylation and then stabilization of this new derivative through resonance, bottom line. I'm just trying to think of when I want to show this video. So there are, in this, not yet, in this TPP complex, in this PDH complex, there are 2 other very important, oh, I forgot about this actually. Hang on, a little bit more about TPP. So I mentioned that this is an important cofactor that we have seen several times already. It was essential to the activity of the non-oxidative decarboxylation of pyruvate in ethanol fermentation. So here we have the formation of acetaldehyde, right, and the release of CO2. We saw it in the transketolose reaction just recently, also dependent on TBP because we're decarboxylating one thing and adding it to another, right? And then finally today, we're looking at it in the context of the PDH complex. So these are some of the principal reactions, 3 principal reactions involving TPP. The other molecules that are quite large, that are essential to the formation of acetyl CoA is lipoate, which has this long arm here. And then the chemistry is going to take place here at this ring with the 2 sulfurs. And we're going to see that it goes, it can be converted to its reduced form and then acetylated with the hydroxyethyl group that has previously been located on the TPP. You'll see in a minute. Just know that lipoate is in part, it's in enzyme 2 basically. And we're gonna pass along, after pyruvate has been decarboxylated, we're gonna pass along this 2-carbon moiety from one coenzyme to the next and one enzyme to the next. It's going to be transferred sequentially from one place to the next in something we call substrate channeling. So, we're going to move this 2-carbon group from TPP, imagine TPP is here. We move it over to the lipoate. And then from there, we actually move it to coenzyme A, which is a huge molecule, right? It has its core as pantothenic acid. It has this ethylamine group here. It has this whole nucleotide type group here. In fact, not type, but a nucleotide group here. Big molecule. In spite of the fact that these molecules are so large, we're always focusing on the part that's doing the chemistry, right? They're long, the size is important here though too. Lipoate in particular has what we call a swinging arm. Can actually move over from enzyme 2 location to enzyme 1 to pick up the 2-carbon group from the TPP. Then it can swing back over and take it to the enzyme 3 and give up that 2-carbon group to coenzyme A. Okay, so I've been talking about the transfer of molecules. And this is what we're looking at here, right? So I want to do a little marking up of this. This is the important part of TPP, this ring, right? This is over here, our lipoate group. And which, as you can see, undergoes a ring opening. And then here we have our CoA, our coenzyme A, which comes in here with this SH group, right? So chemistry here, chemistry happening at this one of these sulfurs here, and of course, chemistry happening at this carbon here in this nucleophilic carbon here on TPP. So the first thing that happens, this is our pyruvate molecule. And this TPP picks up and picks up this 2-carbon moiety. We undergo the decarboxylation. And then this lipoate comes in, it gets oxidized here, reduced rather, here. We open up this ring, and then this group here, our hydroxyethyl group, gets transferred to the lipoate here, right? Are you seeing all that? Okay, so this is hydroxyethyl attached to TPP, right? This shows, the way this is drawn, it's a little funky because this is the lipoate down here, right? And this is the closed ring. So this ring gets opened up, and this group here gets it's what shows up here, right? And yeah, they do this funky kind of, so, and that regenerates, of course, the TPP, which can then just go back over here, right? So this would be like, we're at phase 2 here, right? We've transferred pyruvate to TPP. I mean, we've transferred 2 carbons to TPP, released to CO2. The TPP gives up the this 2 carbon group to the lipoate. Now the lipoate has to transfer the 2 carbon group to the coenzyme A, which happens right here. We're still not done though. At this point, we've regenerated this reduced lipoate, which is then going to get reoxidized so it can undergo another round. And then at this point, we've got coenzyme A. This is our final product, right? And the process, if this is going to get oxidized back to the ring, it has some electrons to give, right? So it's going to donate those electrons to FAD, and FADH in turn is going to donate its electrons to NAD+, and we're going to end up with some NADH. Okay? So it's a whole cycle. Now we're going to really get into the gory detail because why not? Let me see this YouTube video. Anybody have some popcorn? Let's turn off the lights. One thing I want to do. I kind of lost my Zoom ladies. Are you there? Hello? I allowed 2 students to join via Zoom, which I think means they pretend to be here and are not really here. So I will cease to worry about them. Do we have sound? I feel like we don't have sound. Hang on.
Dehydrogenase complex, and we're going to review that mechanism in this lesson. Here's the overall reaction that we're looking at, the conversion of pyruvate to acetyl coenzyme A, a thioester. So we have this sulfur connected to the carbonyl there. And CoA, I just want to point out, is a very large structure. We often abbreviate it with these 3 little letters, and you think it's kind of small, but it's actually a very, very large coenzyme. In this lesson, we're going to discuss the coenzyme thiamine pyrophosphate shown here. TPP is involved in the pyruvate dehydrogenase complex, and we're going to review that mechanism in this lesson. Here's the overall reaction that we're looking at, the conversion of pyruvate to acetyl coenzyme A, a thioester. So we have this sulfur connected to the carbonyl there. And CoA, I just want to point out, is a very large structure. We often abbreviate it with these three little letters, and you think it's kind of small, but it's actually a very, very large coenzyme itself. Let's warm up by discussing the structure of TPP a little bit. This whole portion over here, these two heterocyclic rings, are derived from vitamin B1 or thiamine. Thiamine differs because it doesn't have the phosphate groups here. Instead, it just has a simple OH. Thiamine is a compound that humans cannot biosynthesize, so we need to get it from our diet. Fortunately, plants and microorganisms make this, higher organisms consume them, and it moves up the food chain. And many foods are rich in vitamin B1. Pyrophosphate refers to the two phosphate groups linked together, and these come from ATP. We have two heterocycles in this molecule. The one containing two nitrogen atoms is a pyrimidine. Now, the really important heterocycle in this molecule is the charged thiazole ring. An inductive effect from the positively charged nitrogen atom makes the adjacent proton quite acidic, with a pKa of 18. So in biochemical mechanisms, we'll see that this gets deprotonated, and that's how this can do some pretty amazing chemistry. All right, we want to get into this mechanism, but the pyruvate dehydrogenase complex has quite a few other coenzymes associated with it, so I just need to introduce those first before we get started on our mech. One of the coenzymes that we'll see in this mechanism is derived from lipoic acid. And there's two things I need to tell you about it so that we can understand the chemistry. This is going to become covalently bound to the enzyme via this carboxylic acid, but it is going to attach to a lysine in an amide linkage. And so this carboxylic acid is going to react and be replaced by the amide. So you'll often see this coenzyme called lipoamide because that's the form that it's in when it's attached to the enzyme.
I wanna pause for a moment there because that confused me at first. I mean, sometimes it's called lipoid, sometimes it's called lipoic acid, and sometimes it's called lipoamide. So since this in the active site of the E2 enzyme is attaching to a lysine, it becomes lipoamide. In case you didn't pick up on that. I had to listen to it a couple times before I actually picked up on that.
Also, this sulfur-sulfur bond is pretty easy to break. And so if we open this up, we'll have two SHs. So nucleophiles can attack at sulfur, kicking out sulfur as a good leaving group, and break this bond here. And this results in reduction of lipoic acid, and that'll happen in this mechanism. The other coenzyme we need is coenzyme A, and we looked at that structure earlier. All right, to kick this process off, TPP is going to react with pyruvate. Since this thiazole is going to be the most important piece to do the chemistry in this mechanism, I'm going to abbreviate this in this way, where R is our chain with the pyrophosphate, and R prime is our pyrimidine ring up here. I've shown this in the deprotonated form. This compound with a positive and negative charge on adjacent atoms is called an ylide, and so this is the TPP ylide, generated by deprotonation of this acidic hydrogen atom here. So at this point, with our negative charge on this is going to be the nucleophile in our reaction. And you might be thinking there are two carbonyls that this can attack. Well, carboxylic acids are ionized at biological pH, and so we have this carboxylate. So this already has a lot of negative charge, and it's really hard to attack here. But this ketone has a nice dipole, and there's no buildup of negative charge interfering with this process. And so we're going to get attack at the carbonyl carbon of the ketone. So when we attack here, we need to push electrons up, but in biochemical mechanisms, we can do a little shorthand and have an acidic enzyme residue help us along by protonating this when we push the electrons up. Here's the product of our step. Notice we still have our positive charge on nitrogen, but we used the negative charge to attack here and grabbed a proton. So this is our new OH that we formed from this carbonyl. Now, the next step is going to be a decarboxylation of this group right here. And this is going to occur, as you'll see in a minute, because we can neutralize the ring nitrogen. Right now, we have an aromatic compound, so that's quite stable, even though it has this positive charge on it. And when we push electrons through the system, we're going to disrupt aromaticity here, but we're going to neutralize the positive charge, which is also favorable. So let's look at the arrow pushing for the decarboxylation to see how this works. These electrons are going to come in here. This carbon that is right there is going to now have 4 bonds. So this bond breaks completely. We'll show that, making a new double bond right here. And we have to continue to push electrons, and we can push them onto nitrogen. Now the electrons from nitrogen are going to push back in, making this ring aromatic again. We'll have to push these electrons out and attack something. At this point, we're going to bring in our lipoic acid or lipoamide coenzyme, and we're going to form a bond with the sulfur atom and cleave the sulfur.
Well, this is very good. I feel like, she mentioned that, yes, we're breaking the bond here. I feel like she kind of glossed over the fact that we actually released CO2 here. So yay. And now we're going to move on to,
and when we open up,
of course, to now taking this group here and transferring it to the lipoamide.
Up this ring, we're going to need a hydrogen, and that will come from an acidic residue on the protein. So our arrow pushing will restore the aromaticity. We'll use this lone pair to push in. This bond is going to attack at sulfur. That makes the sulfur bond break. We'll grab our proton from the enzyme and show a lone pair going on to this generic amino acid residue here. Because enzymes catalyze many reactions very quickly, we need to get the coenzyme back to the way it was so it can attack another molecule of pyruvate. So what we're going to do is we need to put a negative charge back on here. We can deprotonate this hydrogen, reform a carbonyl compound, and break this bond to do so. Let's look at how that happens. We will need a basic enzyme residue to do that. This is going to attack this proton. These electrons push down, and when we're choosing which bond to break, we'll put the electrons here and reform our ylide. And we form this thioester here on the lipoamide, which I've just abbreviated with an R here instead of writing out the name of it. Now, this looks a lot like acetyl coenzyme A. We have our thioester, but we need to just switch out our thioester for the S-CoA ester. So we can bring in our next coenzyme and do a trans-esterification reaction. This is an acidic proton. We'll deprotonate it, again, with a basic enzyme residue. So our enzyme uses this carefully positioned base to deprotonate coenzyme A, and this will attack at this carbonyl. Now, we know that we're going to push electrons up and form a tetrahedral intermediate. In biochemistry, we actually save steps often by not showing the tetrahedral intermediate forming. So we know whenever we attack a carbonyl, the electrons come up, they push back down, and then they kick off the leaving group. You should know that mechanism from organic chemistry. But in biochemistry, that whole process is represented with 2 abbreviated arrows. So we can show this just like that. But we have to remember our chemical rules and that we cannot do SN2 chemistry at this SP2 center. So this is just an abbreviation. Some people like to put another arrow to represent that the electrons are coming up, it forms an intermediate and comes back down. But since biochemical mechanisms are so long, just using these 2 blue arrows is completely acceptable. So we've gotten to our final product, acetyl coenzyme A, And I'm realizing with this second arrow, I probably should have grabbed a proton from the enzyme, but I'm not going to show that step. I'll just show the reduced form of lipoamide. Now, I mentioned that all coenzymes need to go back to their original state for the enzyme to be able to complete another catalytic cycle. But we've added electrons to lipoamide, so we have it in its reduced state. FAD can come in, returning it to its cyclic oxidized form. To sum up, TPP is a coenzyme that reacts with carbonyl compounds. It can be deprotonated to form this ylide, which is nucleophilic. And once it does this initial attack step on some carbonyl compound, it's an electron sink, and we can push electrons through the system, neutralizing this positive charge to do things like decarboxylations, and TPP is also involved in some other processes that involve group transfer. TPP here. If you learned something, give me a thumbs up on the way out, and for more chemistry, subscribe to my channel.
Okay. What did Roger and Ebert have to say about that? What do you give it on a scale of 1 to 10? I thought it was good. I thought she did a great job, personally. But have we really hit all the high points here? Let's think about that. Let's go back to our slides. This is, of course, without all the chemistry she was showing. This is the very abbreviated form showing a 5-step reaction. So again, what is it boiling down to? Well, it's boiling down to this. The carbanion moiety of our TPP ring attacks the electrophilic ketocarbon of pyruvate, and we get our hydroxyethyl TPP derivative, as well as decarboxylation. So then one thing that she really didn't talk about is, as I mentioned before, with this lipoic acid or lipoamide or whatever you want to call it, really does have a long arm. And it literally can visit the enzyme 1 site, and we get the opening of the disulfide ring as the ethyl group is transferred onto that ring, and then the other one is just the SH group, right? So the hydroxyethyl group gets attached to one of those sulfurs, and the other sulfur is in SH. Then the swinging arm of this lipoate transfers this hydroxyethyl group once again to the coenzyme A. Then we have the fully reduced form with both SHs, and as pointed out, that has to be regenerated into the ring form. So it donates its electrons to FAD, which generates FADH, and then FADH donates its electrons to NAD+, generating not FADH, but NADH. Okay, so I also mentioned that this is an example of substrate channeling. That just means we're passing this hydroxyethyl intermediate from one coenzyme to another, right? And the final product, of course, here is acetyl CoA. What does this accomplish? Well, it tethers these intermediates to the enzyme complex. That much is obvious. It helps increase the efficiency of the overall reaction, and it minimizes any side reactions. So this is all good and important. Which takes us to the citric acid cycle. Okay, so I know you guys have had, some of you at least have had some exposure to the citric acid cycle, so that's good. And citric acid cycle is actually reasonably descriptive because it is indeed a cycle. We regenerate the oxaloacetate that combines with acetyl CoA in the beginning. So it is a circular process. Sorry, I'm just trying to hide these meeting controls. There we go. Okay, so what's the big picture here? Acetyl CoA, we just took an in-depth look at how acetyl CoA is generated from pyruvate. Well, this combines with the 4-carbon molecule oxaloacetate. And that forms this 6-carbon molecule citrate over here. From there, we go into the cycle with successive losses of carbon. We lose our first carbon here and our second carbon here. And then importantly, we have 4 oxidative steps that generate 3 NADH. So we make an NADH in this step, in this step, and in this step, and we make an FADH here too. So we get 4 electron carrier molecules, and we actually also form a GTP, which is basically equivalent to ATP. So we're going to go through these steps one by one, but this is indeed the big picture. And you know what is highlighted here, the progression through decarboxylations. We don't really see the redox so much here. I mean, we know where the electron carriers are getting reduced, but anyway, we'll go through this one by one. This is the overall thing. Okay, this is actually a second look at all this. It's primarily for your reference, but let's notice it might be worth like looking at these one by one real quick before we address them. We have a condensation reaction in reaction 1, an aldol condensation, right? So we're forming carbon-carbon bonds. We're joining acetyl CoA to citrate. Then we have a reaction that I tend to gloss over a little bit, this aconitase reaction. This is a dehydration-rehydration reaction where we're really just rearranging this molecule, right? So we're moving the OH group, we're repositioning it, and it helps us get to this isocitrate form. So sometimes the citric acid cycle, you'll sort of just gloss over the aconitase step, and you'll really consider, the progression from citrate to isocitrate. That's more, let's say that's more of interest. Once we have our isocitrate, we are ready for an oxidative decarboxylation. So you can really just think of this citrate to cis aconitate and then to isocitrate as a dehydration, water leaving here, and a rehydration, water being added back in here, setting us up, as noted, for an oxidative decarboxylation. Those happen together. We go from isocitrate to alpha ketoglutarate. Then we have a second decarboxylation to succinyl CoA. So we've gone from 6 carbons to 5 carbons to 4 carbons. Everything after this point is going to be, is going to be 4 carbon molecules, okay? One thing I'm noticing here, kind of interesting. I want to go back for a moment. What is this not showing? No, it's showing it. It's just a little hard. There's so much detail on this one, right? I mean, this is great for the detail, but we also want to note the places where we're forming NADH, right? So with each decarboxylation, we're forming an NADH. So that's something to keep in mind. This is a redox reaction, right? This is an oxidation here, and an oxidation here where we form NADH. Then we get our succinyl CoA, our 4-carbon molecule. This is activated by attachment to a coenzyme A molecule, which enables the formation of GTP because we have a high energy bond here, so we can form GTP at this point. We then release that coenzyme. In the process, we release that coenzyme A, and we're left with the succinate molecule. We have another dehydrogenation or redox reaction where we generate our next reduced molecule. And then finally, as we go from fumarate to malate, we have pretty much just a rearrangement here through hydration. And finally, with malate dehydrogenase, remember we talked about this before when we were looking at the malate shuttle. This malate oxidate, this is reduced and this is the oxidized form. So we have another reduction here of NAD to NADH. Okay, for your reference, big picture with details. Where is this all going down? This is going down in the mitochondria. It's a nice space for this to take place because we have all the enzymes, the coenzymes, and the proteins we need for this cycle. This is also where oxidative phosphorylation takes place. So our electron carriers are within good proximity of the whole complex that is the electron transport chain. This is also a location for the oxidation of fatty acids and amino acids to acetyl CoA. And finally, the oxidative degradation of amino acids to citric acid cycle intermediates, because we can, there are different places in the citric acid cycle where intermediates can be fed in and we still get, energy yielding reactions from those intermediates. This just shows a very kind of like basic structure of mitochondria with the outer membrane and the inner membrane. Everything is taking place in the matrix itself. And we can imagine things getting fed in from different angles, the pyruvate, the fatty acids. We don't really see things coming in from amino acids, but that's fine. Yeah, I think that's all we really need to know. Okay, so now we're going to just look a little bit more about the chemistry and some details. I'm not going to go through mechanisms for any of this. At least I don't think so, unless I change my mind for the next class. Okay, so citrate synthase. We need to do this aldyl condensation, right? So this is simply the combining of our acetyl CoA molecule here with oxaloacetate, our 4-carbon molecule. This has a large negative free energy, this reaction. It is a highly exergonic reaction in the formation of citrate and the release of our coenzyme A. This is actually good that it's really, the equilibrium is far to the right because the oxaloacetate concentration is actually fairly low typically. So we want this reaction to be driven far to the right. In terms of the enzyme itself of citrate synthase, the binding of oxaloacetate actually does the good deed of forming a binding site for acetyl CoA. And in the process, the induced fit decreases the likelihood of premature cleavage of the thioester bond of the acetyl CoA, which we need to make this reaction exergonic, right? That's where we're getting the energy to drive this reaction forward. Okay, so that was the formation of citrate. As mentioned, getting from citrate to isocitrate is a two-phase reaction. It's a, well, wait, let me go back a minute, check myself here. Yeah, so we have a dehydration, we lose a water molecule here, and we then turn around and put it right back on. So this is two phases with a conatase, dehydration, rehydration, happening to get from citrate to isocitrate. And then this is our step 3 is catalyzed by isocitrate dehydrogenase. This is something we've seen before. This is an oxidative decarboxylation. And we're taking isocitrate and converting it to a 5-carbon alpha ketoglutarate. If we wanted to analyze the oxidation, we notice that this OH group here on isocitrate is getting oxidized to a carbonyl group right here. And that supplies the electrons for the reduction of NAD+, and in the process, we're losing a molecule of CO2. Note, this is our second exergonic reaction. What was our first? Formation of citrate exergonic, step 3 exergonic. Why am I making a big deal? Well, as we saw in glycolysis, we're going to be talking about regulation at some point. So we want to keep track of what reactions are highly exergonic. Okay, so alpha ketoglutarate dehydrogenase. I think we should probably pay a little bit of attention to enzyme names again, because, we have citrate synthase that talks about, that sounds like we're combining citrate with something else, right? I mean, it sounds actually like we're synthesizing citrate, which we're not. We're combining citrate, but it's bringing two things together. So I think that enzyme name makes some sense. Isocitrate dehydrogenase, whenever we see the name dehydrogenase, we're thinking redox reaction, right? So that's telling a story too. Isocitrate dehydrogenase getting us to alpha ketoglutarate and giving us some NADH. Now we have alpha ketoglutarate dehydrogenase taking the, I'm having a hard time with controlling my slides today. So what's going on here? Well, we have this, this is kind of interesting, right? Because this really does look like something we have seen before. We're basically taking this end of the molecule here, and we're adding in a coenzyme A. So, which should be floating around because we released it not too long ago from acetyl CoA, right? So this is once again a redox reaction and is also a decarboxylation, as well as a forming a high energy bond here, right? So this reaction looks quite a bit like what we saw in the pyruvate dehydrogenase complex reaction. If we look at what was going on there, I mean, we know this went down in a lot of phases here, and we're not even looking at the mechanism here. We're just looking at the overall big picture, but it's actually a very similar mechanism to what is going on with the PDH complex. And note, it is not just alpha ketoglutarate dehydrogenase, it is alpha ketoglutarate dehydrogenase complex. And it is performing almost exactly the same chemistry, except in this case, we're going from a 3-carbon pyruvate to a 2-carbon acetyl CoA, whereas in this case, we're going from a 5-carbon alpha ketoglutarate to a 4-carbon succinyl CoA. I think now would be an interesting time. I've been curious about this, but I haven't had a chance to look it up. Actually take a look. See what this enzyme looks like. I'm just curious to know. Yeah, pretty much the same thing, right? I don't know if we're going to have any pictures here. Look, I mean, that looks, we have an E1, an E2, an E3. I would certainly assume that we have our TPP. Yeah, we have our TPP. We have our lipoamide. Look, can even make this bigger. We have our lipoamide here. We have our TPP. We have our lipoamide. We have our, where's coenzyme A? It's got to be in here somewhere. Here in E3. In any event, do you see it? Oh yeah, here we go. Comes in here. Okay. So really the same game. That's pretty amazing, right? That's pretty interesting. So same chemistry, different day, different cycle. Okie dokie. All right, so now we're down to this. We have this nice activated succinyl CoA compound. What's it going to do? It's going to make some GTP for us. Because why not? We're getting all these reduced electron carriers. We might as well get some GTP as well. You guys need a break? I think we all need a break. I was going to try to get through the whole citric acid cycle, but it's 2:30, so 2:33 actually. So let's, what should we say? How long a break do you need? 10 minutes, 15 minutes? Half an hour.
Right.
Just stop now. Let's come back at 2:50. Almost there, actually.
I have a question.
So do I, for you. You first.
There are some slides, maybe I downloaded, because I downloaded the PowerPoint 2 days ago when you posted all of them. There were a few that
I put a few in just before class today, because I decided to talk more about TPP. I can re-upload.
okay, I was like flipping ferociously. I was like, I don't
No,
know.
sorry.
What's my question?
So my question to you is, you're studying for the MCATs, right?
Mhm.
How, is this relating to?
Yeah, yes,
Uh-huh,
entirely. I mean, I pair my notes with my like MCAT books
uh-huh,
to like drill the concept.
uh-huh.
And it's been really helpful so far.
Oh, fantastic. Good to know. I'm just curious, because I've never touched the MCATs myself.
No.
You don't advise it.
No.
Is anyone else studying for MCATs here? Or thinking about studying for? I know you might be thinking about it. Alexander, what are you thinking about? What? No, I don't mean now right this minute. I mean career wise, like, what are you? You're a chemistry major, right?
Yeah, something in chemistry or
Physics, okay. Don't do physics, do chemistry. What are you looking to? But what are you looking at right now?
Yeah, we all are.
This is more like iterative chemistry. So looks like what we have overall here is a ring opening. Not my, to be honest.
You have to talk to an inorganic chemist for that. Are you doing any research? Who?
Professor Sach.
Barbara? Oh, okay.
She's away for the summer, so we're not graduate students.
Okay. And what kind of project are you working on?
Just basic blood tests, so we got to start class.
Oh, okay. Good though. Few months from what?
A few months from now, I'm actually all on.
Yeah, that's cool. One day at a time. Okay, 250 it is. So where were we? We had just gotten to, well, let's go back and sit and figure out where we were. We went from, we condensed these two molecules, oxaloacetate and acetyl CoA. We got citrate. Then it's not really shown here, but we went to isocitrate. From isocitrate, we went to, this is good because it doesn't have all the compounds. So you can kind of test yourself on, what the intermediates are here. From isocitrate, we went to what? Alpha ketoglutarate. From alpha ketoglutarate, I do believe we went to succinyl CoA. And I think we're about to talk about the reaction of succinyl CoA to succinate, or did we talk about that? I don't even know. We just got to it. Okay. So here we are, conversion to succinate. So we have this activated compound here because we have once again attached a coenzyme A molecule to this in the previous step. And in a slightly oddly named enzyme, succinyl CoA synthetase, we're actually going to in cleaving this bond, we're gonna release the energy needed to form GTP from GDP and PI. So we talked about, I hope, I feel like maybe I glossed over that. Did we talk about substrate level phosphorylation in glycolysis? We did, good, okay. I can't remember what I've said. So this is another example of substrate level phosphorylation. So actually, is it? It isn't. This is a wrong statement. Discard. I'm taking this out. I got to re-upload these slides. Why is this not? This is a good. Why would I call this substrate level phosphorylation? I have no idea. Somehow that snuck in there. Why is this not a substrate level phosphorylation? Over to you guys. Yeah, right. So, oops. We're adding an inorganic phosphate. So this is not substrate. So this should enable you to distinguish, actually. We're simply adding a phosphate, an inorganic phosphate to GDP. Okay. So we can wrap this up by going through steps 7 through 8, which is succinate to fumarate, fumarate to malate, back to oxaloacetate. And this is a series of reactions, 2 of which are dehydrogenations. And whenever you see that dehydrogenation, of course, we're talking about, I've said this before, we're talking about a redox reaction. We're removing hydrogens and forming double bonds. As we are in this reaction, we are oxidizing, and over here, we're reducing. In this middle reaction here, we're just taking fumarate and converting it to malate. And then by adding water, and then in a second dehydrogenation, we're getting from malate to oxaloacetate. So I feel bad. There's a few mistakes here. Sorry about this. Got to just correct this right now. Coenzyme A is not released here. But it's going on. Back. Okay. So none of these reactions here are particularly exergonic, but in the case of malate dehydrogenase, low oxaloacetate will pull this reaction forward by, virtue of equilibrium, and this will regenerate our oxaloacetate for more citrate synthase. One thing that's actually worth pointing out, let me go back to the very beginning here. What do you think the highlighting of this pink highlighting is about? And why does it disappear? Maybe even more to the point, why does it disappear?
That's with the forms, you can't distinguish which side is which because it's symmetrical, so you don't know which 2 carbons there.
Actually, I think you want to go one step earlier. Huh?
Yeah, this is an asymmetrical.
Yeah, that's why if you would lose it here, this one is I would actually say, unless this one has resonance, this is even more symmetrical, right? Because which it doesn't. Well, actually, though, that is in the middle. Yeah, that is symmetrical. Anyway, no, so at this point, we're able to trace this acetyl CoA, right? Molecule through this. And I think in one of the homework problems, I'm not sure. By the way, speaking of homework, I really do promise to do glycolysis metabolism kind of glycogen homework. I'll develop that tomorrow. It's really more for you guys, you know. A, that homework helps you bump up your grade a little bit, and B, it helps you prepare for the next exam. I just haven't, I'm teaching 2 courses this summer. It's been a little hectic. I just haven't had time, to be honest. So yeah, at this point, once we lose this coenzyme A here, we really don't know. I mean, if we drew this differently, we would just show this carboxyl. We don't know which carboxylic acid group at this point is this group, right? We don't know. So, and this is important. Why do we care? Well, imagine you're trying to figure out the citric acid cycle and you radioactively labeled, you know, your acetyl CoA, right? So you can figure out some of this chemistry. Well, that'll work until this point here, but after that, you're going to lose track of this particular portion. Okay, so this wraps things up for the steps of the citric acid cycle. Now let's give some thought to the energy that is derived from all this. So we know that in the course of the citric acid cycle, we make 3 NADH, 1 FADH2, and 1 GTP. So this is important because we know that these reduced electron carriers can lead to the formation of a lot of ATP, and we need to look at the ratio. So each NADH, by donating its electrons to the electron transport chain, drives the formation of 2.5 ATP, and each FADH2 drives the formation of 1.5 ATP. And also keep in mind, if we're starting from glycolysis, all of this is times 2, because we got from pyruvate to acetyl CoA, and we get 2 pyruvates and 2 acetyl CoA from each breakdown of glucose. Okay, so if we do the math, in glycolysis, we got a net gain of 2 ATP and 2 NADH. For the 2 pyruvate to acetyl CoA, we got another 2 NADH, and then all of this from the citric acid cycle. If we use that math of the 2.5 and the 1.5, we end up with a sum total for the complete catabolism of 1 molecule of glucose. We get 32 ATP, which is a number you may have seen before. Okay, now we can consider the citric acid cycle as starting from acetyl CoA, but that's not the only possibility. We can have intermediates that enter the cycle at different points. So in fact, the TCA or citric acid cycle accepts 3, 4, and 5 carbon skeletons. And in the case of the breakdown of amino acids, we can get skeletons such as oxaloacetate. If we deaminate aspartate, if we remove that amine group, we get oxaloacetate. And if we deaminate glutamate, we actually get that alpha ketoglutarate molecule. So the interesting thing is intermediates can enter the cycle, in which case we have an anabolic, a catabolic pathway. However, intermediates can also be siphoned off from the cycle. And for what purpose? For synthetic reactions, for anabolic pathways. So if in fact intermediates are siphoned off or lost from the citric acid cycle, they are generally replenished. And the vocabulary term for that replenishment is something called anaplerotic reactions. These are chemical reactions that may replenish any intermediate that has been siphoned off. So let's consider some of those anaplerotic reactions. Where, oh, where have we encountered pyruvate carboxylase? Anybody? Any idea? It's okay if you don't know at this point, but you do want to know before the next exam. Gluconeogenesis, very good. It is the first bypass reaction for the last reaction of glycolysis, right? So it's where pyruvate gets carboxylated to form what? Oxaloacetate, and then that oxaloacetate, courtesy of PEP carboxykinase, is converted to PEP. Okay, so that's that two-step bypass reaction of gluconeogenesis. Now, there is no undying hard and fast rule that says oxaloacetate has to go on and form PEP, right? And so it could just be used to replenish a TCA cycle intermediate, right? And that reaction is represented here because that first bypass reaction, if it is indeed its ultimate fate is gluconeogenesis, is pyruvate plus bicarbonate, and ATP yields oxaloacetate and ADP. So this right here could potentially replenish the oxaloacetate intermediate, right? Now, oddly, you might notice if it's gluconeogenesis, we go on like this, and that is, so if we're talking gluconeogenesis, this is the first enzyme, this is the second enzyme. Here, however, they're showing this is reversible, right? PEP carboxykinase. So here it's showing PEP actually the enzyme going in this direction, right? And if we look down here, this can take place in particular, apparently, in the heart and skeletal muscle. And we can actually get oxaloacetate out of that as well, depending on what direction, you know, if it's going in the direction, in the opposite direction from what we learned about it. There are other, this is, you know, for mammalian organisms. There are also replenishing reactions for plants. There are other examples. These are all just examples of anaplerotic reactions. We're going to focus on this one, and we're actually going to even look at the mechanism of this one, pyruvate carboxylase. So as noted, we saw this. We saw this in the first bypass reaction of gluconeogenesis. So it is itself a reversible reaction, and it is considered to be the main anaplerotic reaction. Okay. We're going to introduce biotin here as one of these coenzymes. This biotin is a coenzyme for the pyruvate carboxylase reaction. And it's a prosthetic group like we saw with TPP and lipoate and all that. So it is attached courtesy of a lysine residue to the pyruvate carboxylase enzyme. And it's some pretty interesting chemistry, I have to say, because you don't have to know, you should know kind of like this part here. You don't have to know the mechanism, but I would like to take a look at it because I think it's pretty interesting. So what's going on here? Well, we have our pyruvate carboxylase enzyme, and we have our biotin, our biotinyl lysine here. But the first thing that happens actually is bicarbonate reacts with ATP. And in that process, we get a carboxyphosphate that ultimately releases CO2, which is what you see happening here. So we get bicarbonate combining with the ATP and release ultimately of the phosphate to form CO2. That CO2 then attaches itself to the biotin. The CO2 reacts with biotin and we get carboxybiotin. From there, this is what I find interesting about this. CO2 keeps getting released and then getting reattached to something. So here it's attached and we have our carboxybiotin, and then through some arrow pushing here, we get, again, the release of CO2. So we've attached it and then we turn around and we release it. But that sets up this ring structure here to actually attack or interact with a molecule of pyruvate. So it abstracts a proton from this pyruvate and we get a double bond formed in our pyruvate molecule. And we actually get, ultimately what we get from all this is an enolate form of our pyruvate, which then reacts with that same CO2 to form oxaloacetate. So just kind of a cool reaction, right? All this, CO2 seems to, eventually it does get attached to the pyruvate, which is what the reaction is, right? I mean, we know that that's what the reaction has to be. But I find it kind of interesting how the CO2 is getting attached and then released, and then setting up chemistry so that this can react with the pyruvate, and then the pyruvate is then in a form where it can react with the CO2, right? Anyway, as I say, you don't need to know this whole mechanism. You should just recognize the role of biotin in this pyruvate carboxylase reaction. And you know that we have another example here of a prosthetic group, one of these vitamin-derived coenzymes that is covalently attached to our enzyme, in this case to a lysine residue, and has the ability to carry CO2 in such a way that it ultimately reacts with the pyruvate to form the oxaloacetate. Okay, so I think it's been a lot today. I could keep going, but I know, I mean, metabolism, I mean, just general poll, it is like interesting, right? But it's also a lot. But it's amazing. I think it's definitely more interesting than sitting around studying like lipid structures or something, or carbohydrates, but it's a ton of information. It really is. So we'll pick up with regulation the next time, which will be Monday. And then we have a test on Wednesday, as you know. And then we have a test like on August 1st, yes?
So only refer to where you finished from chapter 13.
Yes, none of this that I've covered today, starting from chapter 16, that will be on the next and final test exam form. Regulation of the citric cycle is not bad. It's definitely, I don't think it's going to be as complex as the things we've talked about so far. It's pretty straightforward, not a lot really. So we're really even almost done with chapter 16.